Good morning. Today I will be talking about bioinformatics along with my project about Alzheimer's disease and using bioinformatics for drug discovery. My name is Anirudh Venkatapuram, and let's get right into it. So, what is bioinformatics? Initially, it was the storage and analysis of large genomic data for making predictions, but now it's also used in proteomics, metabolics, and biological systems. Look, bioinformatics is essentially large database management for molecular structure prediction and sequence annotation, along with modeling biological data. So sequences of biological information can reach up to millions of data points, which would take months or even years for humans to analyze and classify by hand. Therefore, bioinformatics takes machine learning algorithms along with all the millions of data points of biology and tries to make sense of all this data using machine learning algorithms. This is a rapidly growing and highly diverse field, so it's difficult to give a concrete definition on what exactly bioinformatics is. The NIH, NCBI, and Oxford Dictionary all have different definitions on what bioinformatics is. Moving on to the general history of bioinformatics, there were four major stages of it. First, it was small-sized databases and fundamental sequence analyzation, and after that, it moved on to sequence analysis algorithms and moderate-sized databases. After that, it switched from solely sequence analyzation to metabolic data and gene expression data analysis, and this made it more applicable for medical and research purposes. In the final stage, which is the stage that we're currently in, bioinformatics is analyzing biological systems and providing the complete picture. So essentially, it's analyzing how cellular level changes affect the biological system as a whole. So for example, DNA microarrays, which are arrays holding thousands of oligonucleotides, were developed to identify gene expression and diagnose chronic fatigue syndrome, hepatitis C, breast cancer, lung cancer, brain cancer, and more. And they do this by making small cellular level changes in the DNA and figuring out how this affects the biological system as a whole and cures those diseases. And the introduction of DNA cloning and recombinant DNA allowed the manipulation of DNA in different species by removing or replacing DNA to figure out the effects. So essentially, they would take a DNA and remove it and see what happens to try and figure out which sections of DNA correlate to which part of the human, which part of certain bodies. Large sequences of DNA nucleotides take up a lot of storage, which led to the usage of bioinformatics databases. Many protein sequence databases and nucleotide databases were created, with one of the major protein databases being SwissProt. For my project, I used the database called Chembl, which is a large database that provides molecules and molecular descriptors along with all other molecules that interact with said molecule. So for example, let's say your molecule is sulfur. Chembl will provide all the information about sulfur along with every single molecule that can create sulfuric compounds with sulfur. Now moving on to Alzheimer's disease. My project involves using Alzheimer's disease and essentially creating a drug or discovering a drug for Alzheimer's disease and then comparing it to the drug that already exists in the market to see how accurate my machine learning algorithm is at creating or finding new drugs. So acetylcholinesterase is essentially the protein which speeds up the rate of Alzheimer's. So what Alzheimer's is it's basically people losing nerve cells. So people with Alzheimer's lose nerve cells as the disease progresses. And this is known as Alzheimer's. So in normal people, acetylcholinesterase is a protein in people that don't have Alzheimer's. Acetylcholinesterase is a protein that stops the signal between a nerve cell and a muscle cell. So basically, nerves communicate through muscles using neurotransmitters, and they can tell the muscle to either contract or loosen up and expand. So acetylcholinesterase destroys those neurotransmitters after they have delivered the message to prevent any jumbling up of messages. So acetylcholine is a neurotransmitter that carries signals from nerves to muscles, and once it passes the signal, it has to be destroyed to prevent later signals from being mixed up. So in order to essentially cure the symptoms of Alzheimer's, what scientists are currently doing is that they're inhibiting acetylcholinesterase to raise neurotransmitter levels so that it can strengthen the nerve signals that remain and cure the symptoms of Alzheimer's. Now moving on to my machine learning experiment with acetylcholinesterase. So for my project, this is basically an experiment to see how accurate a machine learning model is at finding the drug to cure the symptoms of Alzheimer's disease. And the result is to figure out how closely a machine learning model can come to an existing drug, which inhibits the protein acetylcholinesterase from breaking down acetylcholine, which is the neurotransmitter that leads to the symptoms of Alzheimer's after it's broken down. So starting with part 1 of my project. So this project first requires me finding a single protein target from the Chembl database. And technically, you could use this process for any disease as long as it's caused by a single target protein. And also, if at any point during this presentation you come across words that you do not understand, please wait until the glossary section at the end of this presentation. Thank you for your patience. So what I have to do next is to find and retrieve bioactivity data for the single target protein, which is reported as an IC50 value in nanomolar units. So then I would have to sort the compounds to either active, inactive, or intermediate compounds. So bioactivity data is in the IC50 unit. So compounds having a value of less than 1,000 nanomolars will be considered as active, and those with greater than 10,000 will be considered inactive. Anything between that range would be considered as an intermediate compound. So I would have to take all the bioactivity data, all these values of like 1,000 to anywhere above 10,000, and then I would have to combine the 3 sets of data of active, inactive, and intermediate and place them as columns inside a data frame, and then combine the data frames. Now moving on to part 2 of my project. This is where I calculated Lipinski's descriptors. So again, if you don't know what Lipinski's descriptors are, I have included a glossary section at the end of this presentation, which I will go over. So I first had to calculate Lipinski's descriptors, and the Lipinski rule states the following. Molecular weight has to be less than 500 Dalton, octanol water partition coefficient, or log P, has to be less than 5, hydrogen bond donors has to be less than 5, and hydrogen bond acceptors has to be less than 10. If a drug violates more than one of these rules, then it will not work on humans because it'll just get diluted with water and it won't have any effect. Therefore, it needs to follow all these rules to have some sort of effect on humans. So I calculated the Lipinski descriptors using the Python code sourced from that link. And here's the actual code for defining Lipinski's descriptors. So what I did was I put this code into a data frame and concat that, and I had to concat that with the original data frame from part 1. So after this, we would have to convert IC50 to PIC50. And this is to allow the IC50 data to be more uniformly distributed. And so essentially what I did was I converted it to the negative logarithmic scale, which is essentially negative log base 10 of the IC50 value of anywhere between less than 1,000 to greater than 10,000. I mean, anywhere between 1,000 and greater than 10,000. So this custom PIC50 function will accept a data frame as an input and will convert IC50 from nanomolars to molars by multiplying the value by 10 to the power negative 9. And it will take the molar value and apply negative log base 10. And on top of that, it will delete the standard value column containing IC50 and create a new PIC50 column in place of the IC50 column. And after that, I would have to remove the intermediate class from part 1 to simplify the data frame so that there are only active and inactive classes. After this, I would have to run descriptor tests to determine the statistical analysis through 4 different tests: the Mann-Whitney U test, log P test, the number of hydrogen donors test, and number of hydrogen acceptors test. And after that, this results in 4 separate graphs, which each of the above tests being on the y-axis and the bioactivity class on the x-axis to determine statistical significance between the active and inactive compounds. So in this slide, as you can see, there are 4 separate graphs. And the first graph is the Mann-Whitney graph, which shows statistical significance because there's a significant difference between the medians of the Mann-Whitney U-test and the Mann-Whitney U-test for the inactive class and the active class. For the log P test, there is barely any statistical significance since they're very similar. For the bioactivity class for the number of hydrogen donors test, there is definitely statistical significance, and same thing applies to the number of hydrogen acceptors test. Moving on to part 3 of my project. So this is where I would have to calculate the Pedel descriptors. So I would have to take what was downloaded in parts 1 and 2 as a CSV file, and that will be used to build a regression model for machine learning. So first, I would have to download the Pedel and load the bioactivity data, and then I would have to calculate the fingerprint descriptors. So again, if you don't understand some of these vocabulary terms, I have included a glossary at the end of this presentation, and I will read that out loud. So I will have to sort the descriptors into 2 data matrices, the descriptors output as the X data matrix and the Y data matrix having the IC50 values, which I first converted into PIC50 and placed into a matrix. So the X data matrix has the descriptors output, and the Y data matrix has the PIC50 values. So then I would have to combine the 2 data matrices and download the dataset as a CSV file for the model building. Now moving on to part 4 of this project. So this is where I started with the regression model building. So I'd have to build a regression model of the acetylcholinesterase inhibitors. So essentially, from Alzheimer's, in order to cure the symptoms of Alzheimer's, we need to inhibit the protein known as acetylcholinesterase to raise neurotransmitters. So I would have to build a regression model of the acetylcholinesterase inhibitors using the random forest algorithm of the dataset downloaded in part 3. So I would have to take the dataset and split it back into the X and Y data matrices, and then examine the data dimensions and remove low variance features to make the dataset easier to read and understand. And then I would have to split it between an 80-20 ratio and build a regression model using random forest. And then I programmed it to generate a scatter plot with a dataset based on experimental and predicted PIC50 values. And then moving on to part 5. These are comparing the machine learning algorithms for the regression models of acetylcholinesterase. So I used an open source Python library called LazyPredict to compare various machine learning models that already exist of a dataset. So I compared the R-squared values and the RMSE values to figure out each machine learning model's accuracy. The higher the R-squared value, the more accurate it is, and the lower the RMSE value, the more accurate it is. So essentially, when looking for machine learning models that are accurate, you want to find high R-squared values and low RMSE values to get the most accuracy. And now, up until now, I have been describing the methods that I used for my project, and now I'll be describing the results. So the target protein that I used was acetylcholinesterase, as described before. And through the method from everything described in part 1 to part 5, I have obtained the following results. So I obtained my own X and Y data matrices for part 3, comparing the inputs with PIC50 values. And to the side over here is the graph of the predicted PIC50 value versus the experimental PIC50 value from part 4, showing the regression model using random forest. So since it's basically a 1-to-1 ratio, that means it's pretty accurate. And for the ML algorithms, I noticed that the machine learning algorithm called decision tree regressor showed the highest R-squared value and the lowest RMSE value using, as you can see from the two graphs to the right. And this makes the decision tree regressor algorithm the most suitable for understanding acetylcholinesterase inhibitors and prevent the symptoms of Alzheimer's disease. So, using the machine learning algorithm known as decision tree regressor, I have used it and compared the predicted PIC50 values, which are being used to cure the symptoms of Alzheimer's, and compared it with my experimental PIC50 values, which is the decision tree regressor machine learning algorithm. And they have a ratio that is close to 1-to-1. So as you can see, as the experimental PIC50 increases by 1, the predicted PIC50 increases by about 1. So what this means is that as the PIC50 of the actual drug increases by 1, the experimental PIC50 also increases by approximately 1, which means that the drug discovered by the machine learning algorithm is relatively close to the real drug which currently cures the symptoms of Alzheimer's. So essentially, this entire project was checking to see if a machine learning algorithm could mimic drug discovery and figure out how exactly pharmaceutical companies use machine learning algorithms for drug discovery like this. Thank you. Now moving on to the glossary, which I stated before. So first, the Chembl database is a manually curated database of bioactive molecules with drug-like properties. So it brings chemical bioactivity, and genomic data to aid the translation of genomic information into effective new drugs. And then IC50 is called the half maximal inhibitory concentration, or IC50, and it is the most widely used informative measure of a drug's efficacy. So this indicates how much of a drug is needed to inhibit a biological process by half, thus providing a measure of the potency of the antagonistic drug in pharmaceutical research. And then moving on to Lipinski descriptors. So Lipinski in 1997 was based on the observation of orally administered drugs and relatively small and moderately lipophobic molecules. He came up with a rule that describes molecular properties that are important for a drug's pharmacokinetics in the human body. And this includes their absorption, distribution, metabolism, and excretion. And he came up for this thing called ADME, which is basically absorption, distribution, metabolism, and excretion. So Lipinski's rule of 5 predicts that poor absorption or permeation is more likely to happen when there are more than 5 hydrogen bond donors, more than 10 hydrogen bond acceptors, the molecular weight is greater than 500, and the calculated log P is greater than 5. So that means when all of these are more than when it's more than 5 hydrogen bond donors, more than 10, more than 500, and more than 5, that means the drug is more likely to just be diluted in water, and then it goes to waste. So by following these, by having less than 5 hydrogen bond donors, less than 10 acceptors, less than a molecular weight of 500, and a lower log P value than 5, you can make sure that the drug actually does something to the human body and doesn't just get washed away by water. Now, the Mann-Whitney U-test is a non-parametric test of the null hypothesis that for randomly selected values of x and y from two populations, the probability of x being greater than y is equal to the probability of y being greater than x. So essentially, it's a statistical test to measure how statistically significant the results are. And then the log P test is also the Mann-Whitney U-test, log P test, number of hydrogen donors test, and acceptors test are all tests that measure statistical significance between the active and inactive classes. So the log P test takes the log base 10 of the partition coefficient, and the partition coefficient measures how much of a solute dissolves in the water portion versus an organic portion. And it is an important measurement of the physical nature of a substance and thereby a predictor of its behaviors in different environments. The log P value provides indications on whether a substance will be absorbed by plants, animals, humans, or other living tissue, or be easily carried away and disseminated by water. So the number of hydrogen bond donors is the number of hydrogen bond donors that the active drug has created by the machine learning algorithm. And this value should not be greater than 5. And same thing applies with the hydrogen acceptors, except the value cannot be greater than 10. And then Pedel is a software for calculating molecular descriptors and fingerprints. The software currently calculates 797 descriptors and 10 types of fingerprints. These descriptors and fingerprints are calculated mainly using the chemistry development kit, or CDK. And finally, acetylcholinesterase is an enzyme that causes rapid hydrolysis of acetylcholine and its action serves to stop excitation of a nerve after a transmission of an impulse. So essentially, it just destroys neurotransmitters. And that's about it. These are the Google Colab research PDFs that I have used in my project on acetylcholinesterase. So the first two are examples showing that this machine learning algorithm sequence analysis can be used for basically anything. And then the part 3, part 4, and part 5 are all my database, all my PDFs, which I used for my machine learning project. And that's about it. Thank you.